Good afternoon. I would like to start by saying thank you to the organizers of this workshop for the opportunity of explaining a little bit more of the information we have in our poster called Transgeneration, Epigenetic Aspects Driven by Maternal Phenotypes in Dairy Cattle, a Co-methylation Network Approach. I want to start by briefly introducing myself. My name is Adrian. I'm a PhD student from Spain. I'm based in Madrid in a research institute called INIETHESIC. But right now I'm pretty far away from home. I'm doing a short research stay in Brisbane in the CSIRO. So you're probably wondering why am I 18,000 kilometers away from home? The answer is pretty straightforward. It's to learn a set of key skills that are really hard to develop in Europe. So for example, how to hold a koala. But that's actually pretty easy to learn, and it only took like 15 minutes. So I still had 4 months here. We talked with the people from Spain, people from here, and we decided to explore if a cow phenotype could be altered in her offspring epigenotype. We call epigenotype of the sum of all the methylation marks in the genome of an individual. So I'm going to be using this term in this presentation. And this is what we got. This is the Co-methylation network. You're gonna have to wait a little bit for the explanation of it. I'm gonna be guiding you through the process of how did we get there, what does it mean. So our initial idea comes from the fact that primiparous cows, this is cows that are in their third pregnancy, do not lactate during the pregnancy, during the gestation of the new calf. But multiparous cows that are cows that are in their second pregnancy or more, in this case, their lactation peak will be overlapping with the pregnancy of the next calf. So there's going to be a really huge negative energetic balance that it doesn't really matter how much we try to feed the cow. The energetic balance is going to be always negative. She's going to lose body weight. So the calf is going to be fighting or competing for nutrients with the mammary gland. Our hypothesis is that cows born from multiparous cows are going to be subject of a metabolic stress that is not going to be happening in the cows gestated by primiparous cows, just for the fact that these primiparous cows are not lactating. So we posed ourselves 2 questions. The first one was, is the methylation profile really going to be different in cows from primiparous cows than in cows from multiparous cows? And for that we proposed to do a differential methylation analysis. And the second question was, could a cow phenotype, the mother phenotype, affect or modify the calf epigenotype or the methylation profile of the calf? And for that we did a co-methylation network, the one I just showed you, based on a PCIT algorithm. Our material are pretty straightforward too. We have cow born from 2 different group of cows. We have 3 cows born from non-lactating or primiparous cows, 3 cows born from lactating or multiparous cows, and we took blood samples from each one of them. So we have 6 blood samples. We perform a DNA extraction using this kit here, the Monarch. This is a kit specifically designed to obtain heavyweight molecules, long molecules of DNA, because we are sequencing those samples using nanopore sequencing. It relies on long reads. It basically works. It's based on the disruption of electric current in these spores that you can see on the left-hand side of your screen. When the DNA passes through the pore, it's going to create this disruption. Each one of the bases has a different electric profile, and the methylated cytosines have a different profile of that of the unmethylated cytosine. So we can translate these straight lines that you can see here, that are called the squiggle, into the actual base sequence, into the nucleotide sequence in a process known as base calling. For that we use a GridION sequencer. We base called using GAPI. We called methylation. We obtained the methylation frequency using a software called F5C that it's a GPU version of Nanopolis. It's a little bit faster, and perform the differential methylation analysis using the SS and R package. We fixed a delta that is the minimum difference that needs to be in the average, in the estimate of the methylation average in each one of the groups. A false discovery ratio of 0.05 and a distance to the transcription start site needs to be lower than 50,000 base pairs. And then we also do this BCIT algorithm and represent the correlations in cytoscape. This algorithm estimates the correlation between two genomic regions, or the genomic region with the calc with the phenotype of the model. So what we obtained in the differential and methylation analysis was that there are actual differences between these groups of animals. We found almost 54,000 differentially methylated cytosines, and 54% of them were hypomethylated in cows born from primiparous cows. And we also found 2,355 differentially methylated regions, being 58% of them hypomethylated in cows born from multiparous cows. So then we split the original dataset, taking into account these regions. So we split it into regions that were associated to hypomethylation in primiparous cows, hypermethylation in primiparous cows, and also two datasets of the differentially methylated cytosines. We are always using the cows born from primiparous cows as the reference. So if we say that region or a cytosine is hypomethylated, it's that it's going to be less methylated in the cows born from primiparous cows than in the cows born from multiparous cows. But what we were able to observe is that the hypomethylated regions and cytosines seems to be located in the promoter regions of the genome of these animals. That's what you can see in the image in the top, and the one in the bottom. In the one in the bottom, and the one you can see in the top is the distance of these cytosines and regions to the transcription start site. So what I can interpret from here is that promoter region seems to be hypomethylated in the cows born from primiparous cows, which can be translated to an increase or a higher gene expression when compared to the genes associated to this region of cytosines when compared to the gene expression of the cows born from multiparous cows. We extracted the list of genes associated to these cytosines or regions. We did an enrichment analysis to understand what's going on in there. So what we could see is that genes involved in cell migration, locomotion, and cell fate commitment were hypomethylated in the cows from primiparous cows. Genes involved in morphogenesis of several different tissues, systems, development, and signaling activities were hypermethylated in these cows from PP. And then we extracted the list of these genes and use it to represent the match in the network. I'm going to be getting into that in a couple of slides. So what we can say here is that there are indeed differences in the methylation profiles of cows born from primiparous cows or non-lactating cows and cows born from lactating cows. And knowing that, we wanted to see if there is also a possibility of this molded phenotype explaining part of the difference in the methylation profile of these regions or cytosines. So we constructed the methylation network that you can see here and you have already seen. You can see that there are different sizes of nodes that are the dots or the bolts, however you want to call them. The bigger dots are the genes and regions associated to the genes that we found enriched in the differential methylation analysis. And the one in the outside the network are the phenotypes of the mother. We constructed this network breaking the genome of the animals in windows of 500 base pairs. We estimated the mean or the average methylation profile of each one of the windows, and then we run the BCIT using the information in each one of these windows. So what you can see here is the correlation between the methylation frequency of each one of the regions, one of the poles, and the phenotype of the mother of the calves. So we also did an enrichment analysis to see if the genes that we have found correlated were also related to embryo development or morphogenesis processes. That it was like that. We found that a lot of these genes were involved in developmental processes, in multicellular organism development, anatomical structure development. So are genes that are involved in the correct or ensuring a good development of the embryo during the gestation of the animal. And there was indeed a difference in the methylation profiling of these two groups. So going back to the network, what you can see here is that the nodes are colored according to the methylation status of that region, taking into account the results of the differential methylation analysis. So in green are going to be genes hypomethylated in cows born from primiparous cows. In pink are going to be genes that are, or regions that are hypermethylated, and in brown the dance phenotypes. And the edges that are these arrows connecting dots are going to be colored according to the correlation they denote. Blue for positive, red for negatives. But it's actually kind of hard to get any information out of this. So what we did is to select these 4 phenotypes here: meal yield, number of parturition, number of lactation, and somatic cell count. And we took the first neighbors. This is the nodes that are directly connected to these phenotypes, to these phenotypes, and got 4 different individual networks. And I'm going to be taking meal yield as an example here, just to make this not really tense, and so you can get the idea of what we were trying to do. So when we look at these individual networks, what we have to see that there are actually 6, but let's focus on 4 different combinations. We have these bluish greenish nodes. That's going to mean that they are hypomethylated in calf born from primiparous cows. And in pink, the regions that are hypermethylated. But we also need to take into account the color of the edge. So it's going to be blue for positive, red for negative. And we always need to analyze this network from the middle to the outside, because it's the phenotype of the mother that it's going to be affecting the epigenotype of the calf, or the methylation profile of the calf. So we have to read them that way. We're going to be taking palm D as an example here. As you can see, it's hypomethylated in cows born from primiparous cows, and it has a positive correlation with milk yield. This gene has been described to have an association with milk production and to be involved in milk yield. So what we can understand from this image here is that the more milk produced by the mother, the more methylated that this gene will be. So it makes more sense that it is hypomethylated in cows born from primiparous cows, as these cows do not have milk production. If this effect is not reversed during adulthood, cows born from multiparous cows will have a reduced gene expression of this gene. So what we can try, what we are trying to explain here is that the milk yield, the phenotype of the mother, is going to have an impact on the future phenotype of its calf through the methylation status of this region. To finish, I want to give some take-home messages. Is that blood is a non-invasive technique that can be used to epigenotype animals for methylation marks associated with reproductive traits. That we have found more than 50,000 differentially methylated cytosines associated with metabolic stress. Our networks also indicate that the phenotype of the mother is correlated with the methylation frequency of different genomic regions of their offspring. We know that example is a really reduced sample size, so we are working on increasing this sample size to ensure that the methylation marks can be used as biomarker to detect metabolic stress. To finish, I would like to thank all the people here. I would like to thank the people back home at INEA, the people here at PSRO for helping me and guiding me through the process of understanding how this network works. I would like also to thank CONAFE and FEFRIC, that are also increasing associations in Spain, and SATSAMER, that it's the farm that allowed us to take blood samples from their calves. And thank all of you for your attention. If you have any question, I'm open to them.